Better to be wider than not to be.
So this is usually go to meeting, but it usually doesn't affect us at all. I just, at the end of the talk, I check in the chat box if there's any questions.
Okay.
You're good to go?
So people are going to be online, because I tend to ask questions also.
Ah, well, they will.
So-
Yeah, they're usually muted. Okay.
Yeah,
We
by
don't have
default.
that many people online, to be honest.
Okay.
What we can, by default, we tend to mute them just in case they're eating or doing anything else. If you would like, I can unmute everybody.
They can always send it by chat. Is
Yeah.
Yes.
that going to be monitored at all? or-
I can.
Yes.
Oh, you can. You just have the box open. And then you
So
can see.
really, I like it to be interactive if possible. So anytime anybody has a question, we can stop and address the questions then.
Perfect.
So let me do this. Let me do it like
I
this.
tend to talk a lot.
Yeah.
And I think that there are more slides here that I can legitimately cover. So,
When we reach like 5 minutes till, I'll be like,
yes,
eyebrow,
that'll be very, very useful.
zero.
Well, the other confounding thing is I can talk very fast.
Ah, so we can be done at
So,
12:40.
We could be.
All right, so what I'm going to do, because the chat box is too big, it's going
Okay.
to end up covering the
Ah,
folks
okay.
here. So I'm putting on chat bubbles. What that means, it'll tell you somebody just sent a chat and did something, and then all you have to do is just open this
And it will pop up over
and
there.
look at C right here under chat and just see
Gotcha.
what it is that they had to say, and then you can minimize it back down.
Yeah.
Okay?
That work?
Does that work? Cool deal.
Great, thank you.
You're welcome.
So you've been here about a year, I guess, huh?
April 4th was a year.
Yeah. Wow.
It does. It doesn't seem like, well, in some ways it seems like not long enough, and in other ways it seems like a bit intense.
Yeah.
So,
So you were at UK before?
yes.
So what location were you at?
King of Prussia, which is right outside Philadelphia.
Oh, okay. Mm-hmm.
So that was
Like a mall.
What was that?
Like a mall.
Yes.
That's the only reason I
You know the mall.
King of
That
Prussia.
was the blank expression on my face. You know the mall. Okay.
Foreigner, you know the highlights of the country. Grand
Well,
Canyon Mall.
My mother gives all directions by locations of Walmart and McDonald's.
Very practical.
Yes.
Mm-hmm. Yeah. So how's it been from Birmingham?
I'm originally from the South, so Florida, Florida.
Okay.
So
Yeah. This is the.
Birmingham does not have critical mass of research going on that I see in the Northeast.
Yeah.
And in
So-
Yeah.
so from that
So your
standpoint,
grant,
I realized
you submit a couple
for
of
me, it's
right?
fine.
Yeah.
But when I'm looking to hire
Review.
Yeah.
just talking to people.
Mm-hmm. I can see that. Yeah.
I have enough,
Yeah, there's good things and bad things
I
about it.
was,
Good thing is,
I did more than some time to get more data before I submit.
bad thing is, it's something's hard.
But I think I'll put the video,
I hear the
help
same.
advice, one way is right plan, okay? Will be based on your, the
Big
comments, okay?
information interview early, especially if they're not coming back.
Yes.
Now, that's.
One of the things that I had a position for a toxicologist that was open for, it was over a year. It was actually open before I came, and it took that long to fill
Yep.
it.
Mm-hmm.
We got a great candidate.
Yeah.
But how in the end we found out somebody was just selling them on quality of life here,
Yeah.
not on, this is going to be a big thing for you.
No, yeah.
Just really
So,
Yeah.
So
So
what
which
he
institute?
is,
is I institute, right?
and
So,
is
so
ready to.
National Institute of, National Institute of I, is that right?
Easy, but
I think
he's
you get your
not done for himself.
Yeah, yeah.
Oh,
Mhm.
too much.
Like a younger individual.
Yeah.
So getting him in here.
Is that NCIR now have a lot of money for
I can see that.
drug discovery?
Yeah, and our cost of living's low, so sometimes it helps. Chemical.
Yes. 90% cheaper here than
Yeah.
Philadelphia.
30% net.
No, I don't
Yes.
think they will from you sometimes.
Yeah. When I was a postdoc, this guy in San Diego tried to recruit me. I was kind of as a postdoc or maybe, and he was going to pay me the same salary as a making in Birmingham. I'm like, that's not going to work for me. He said,
No,
well, but everything nice here is free. You can walk on the beach for free. The sun shines over them. I'm like, yeah, you're not selling it. That's
No,
not gonna, I still need to
no.
rent an apartment and buy groceries.
Exactly.
And what you're offering to pay me.
It was a peanut butter and jelly sandwich.
Yes, I'm close. Yeah.
Different stages of life,
Yeah.
different things are important.
Mhm. Yeah, if we would have asked 1 or 2 years earlier, it would have been different. I think, but it was about the position of faculty here. And
Yeah.
That's what it is. Now, go with Mark Pseudo and Mark Rivera to bio. In June is coming up. So, really told Mark Rivera to start making the social program.
So Mark just sent me, there's a grant that he is applying for.
Yes, for NIAID.
Yes.
Yep.
So you're going to be part of that?
We talked about it yesterday
Okay.
because I read it and I think it would be a great fit because I think you need to hit at least 2 of the 5 main areas that they want you to have expertise in, and I think you can hit at least already can hit 2, but with you it would be 3. So I think it sounds like that.
So that's good. Yeah, he just shot me an email asking if I would be willing to be part of that.
Yeah.
We haven't had a chance to talk about it yet.
Yeah.
So looking at it, I think it would be a really good fit for us.
And it's multi-year
Yeah.
possibilities, so.
For that, IPIQ approach, we do a lot of.
Yeah. Yeah, it came on our radar because we got the big antiviral U19, and because it was not virus specific, it was more, we have the technology that if they can have a project, they can just shut it away.
Yeah.
So, especially now with you have a track record now, at least some research to work with a lot of different antiviral biologists, so you know speak the language, and to deal with prima donna, personalities.
So,
Everybody has. Somebody asked me the other day, what I did for a living. There's a lot of ego massage.
oh,
In a good way. Especially the U19 nephrologists we work with, they're all used to being the smartest and all-knowing person in the room, and then learning how to do drug discovery, all of a sudden they figure out that they actually don't know the answer to everything.
well, the ones who actually figure that out, if you send them to me, because one of the biggest problems I face is really smart biologists
Yeah.
who don't really understand.
Yeah.
But
I think they do.
they think they do. So they come up with this very detailed plan on exactly what they want to do to achieve their goals. What don't meet the requirements. And really, and then trying to get them to understand that is sometimes.
Yeah. All right. I can see that.
So I'm say is really man at Stanford.
Yeah.
But the clinical plan that he's putting together is just
Yeah.
so.
Yeah, and then the humility is lacking.
Yeah.
That's not a good thing. No. At least we have Richard, who's my red boss, who's the PI for U19, and he's a big shot philology guy himself, so he can kind of usually be the intermediary to really easy to get to that. Usually there's no issue. Then you can elevate to my level of smoothing the open. If that doesn't work, then we okay, this is why, the chemist asked me for this, not because he's wanting to do more work, but this is the reason why I need to keep your information. You shouldn't have to explain that, but sometimes you do. All right, let me see who's back here still. Wait, perfect. Hey everybody, thank you for coming on this beautiful day. I hope you all signed in. If you haven't yet, please sign in on your way out. Every week, I keep forgetting that we have sign-in sheets, so maybe by the last lecture I'll remember for sure. So today we have Paul Bushkit as our speaker. He's never spoken to us before yet, so we're very excited that
Be
he's
gentle.
exactly. He's been a year or so at Southern Research as the head of toxicology operations there. I don't know exactly what your formal title is, but that's kind of what it explains it. He came from GSK before that, so has a lot of experience in this field. For the people online, let me open this up real quick to see who is here. He said he likes to keep things interactive, so if he asks a question, feel free to also when you're online, put in the chat box an answer and it will pop up on our screen. And then maybe I can actually open this at some point. Oh, there we go. Yeah. So I will see your question in the chat box and then we'll get to that way. I'm going to minimize it for now. So with that, there you go.
Real quick. So I need bifocals, but I refuse to get them. So there we go.
There you go.
Okay. So I may have to pop these on and off. To see you, I have to have glasses. To read this, I can't hear them. So you did say that I came here from GSK. I've been here slightly over a year at Southern Research. I am the Director of Toxicology and Pathology Services.
See, I would not remember
That's
that.
the full mouthful.
Yeah.
Tox is how we refer to it. At GlaxoSmithKline, I was there for 12 years. I started out as a study director in regulatory studies in reproductive toxicology. I then went on and I was the safety assessment representative to multiple drug development teams related to antibacterials and cancer therapeutics. So in that realm, what I did is I acted as a consultant, peon, runt in the field, and also shield to the teams against the regulatory departments, all related to safety though. And that went on for about 10 years in that role. So today what I want to talk about just in general is the role of toxicology in drug development though. So I have a lovely little phrase down at the bottom there. Does anybody read Latin?
Little rusty.
Little rusty. I took Latin for several years. I couldn't translate it myself. I had to go on Google. Okay, I pick on people too, by the way.
No, we don't speak on you.
That's okay. It's okay.
The dose is the point.
Yes, sir. Did you know that from reading this, or did you already just know that?
Do it for reading, pick up 2 words, yes.
All right, perfect. So the dose is the poison. That pretty much describes toxicology. Toxicology, by definition, is the study of poisons. So, a little bit wordier phrasing. It's this toxicology study of adverse effects of chemical, physical, or biological agents on people, animals, and the environment. We're not going to focus on the environment. We're going to focus on actual deliberate delivery of an agent to people. Well, that's the goal in the end. I don't do it to people, I do it to animals. A toxicologist is a scientist. Some people sometimes forget that. We actually are scientists. We're trained to investigate and interpret, that's a key phrase here, what are the effects of the agent on a living individual. Whether that is in your animal model or in humans, a toxicologist still assists in both aspects of it. Factors that influence the chemical toxicity. Dosage, 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 dosage. That is the most important thing. Paracelsus, he actually coined the original phrase, which was then later adapted to the Latin phrase, which we saw, which is the dose of the poison. What he actually wrote was solely the dose determines that a thing is not a poison. Yeah, I said that right. So one of the things to keep in mind is anything and everything around you is in fact a poison. The air you breathe is a poison. The water you drink is a poison. It's all about dose. You eat too many green beans, you're going to get sick from it. So toxicology is following a natural phenomenon of dose effect on a living organism. So what do we do in toxicology testing? Really in relationship to drug discovery and delivery. We need to prove new drugs are safe before administration to humans and then before clinical trials. How many people think that is actually a true statement? Raise your hands. Is that true?
It feels like a trap.
It is a trap. You're very smart. What is the trap? Who can point out the trap?
Well, in fact, we have to show that it's not toxic, let's say, opposite.
Perfect. Safe is not a realistic goal here. If everything around you is toxic at some dose, how can anything be truly safe? It isn't. You have to define what it is. What we have to do is we have to define hazard identification. So what is the critical aspect of that poison response? So we also have to develop what is known as a risk assessment. Those are two separate things. Another quiz. Who can I pick on? You already answered. All the way in the back. Lovely lady sitting in the chair. What do you think is the difference between a hazard and a risk? Which is worse?
The risk is a chance of something like.
You know, you would think that. And I actually asked one of my fellow scientists this morning about that at Southern Research, and they gave the same answer that you did, which unfortunately is not right. So a hazard is a potential.
Yeah.
A risk is you're right in the water with the shark, and there's real risk associated with it. But the hazard is the identification of something that is a potential problem, but it's not of immediate threat. Doesn't make sense? It's a definition for it. You just have to go with it at this point. It confuses pretty much everybody until you're in the field for a while. So, if as a toxicologist, the point is to prove that drugs are safe, in drug development, where does that come into play? When do you start with toxicology testing? How long does that go on? What is the responsibility for toxicology in the overall drug development scheme? So I know that you guys have gone over in generic terms sort of drug development, basically from the identification of a potential disease and target and all the way to market and what happens beyond. When does toxicology testing become important? Sorry?
Preclinical.
Preclinical is a very broad area.
Will you have,
Before anyone.
will you have a big compound?
Okay? Anybody else? I would say when you have a lead compound is true, but it's also an old school way of thinking that is not along the lines of current drug discovery. Toxicology actually starts when you identify the disease and the target that you want to go after. When does it end? Never. It never ends. Even after a drug is on the market and it's been in humans for a while, something new pops up. You've got to go back and do toxicology to test it. So, with that said, going back to definitions, hazard and risk, doesn't make sense. It's a definition, we just got to roll with it. Preclinical, clinical, toxicology is actually considered non-clinical. And the reason for that is, yes, you do some toxicology preclinically, but believe it or not, the bulk of real in vivo toxicology work is done after people are already seeing that compound. Definition-wise, it's no longer preclinical, you're now in the non-clinical area. So, here we go. Yeah, I think I better slide forward. Here we go. So in the discovery phase, that's when you're really looking at identifying the disease you want to go after. What is the molecular target? You're starting those initial medicinal chemistries in order to get sort of test compounds. You're going to then go through millions, thousands, however much your compound library is, to sort of sort out what are the chemical series that you're going to be going after. Toxicology actually plays an important part here as well. It's not necessarily the in vitro assays and it's not the in vivo assays, but what it is a trained toxicologist looks at data differently than a biologist does. A biologist who is looking at a disease and looking for a potential therapeutic target for that is very focused on disease and how that molecule is going to affect the disease state. They don't necessarily take into account what happens in a normal individual in the rest of the body and the rest of the tissues. So an entire field of toxicology that is growing enormously in the field right now, it's called computational toxicology, which goes back and looks at the molecular pathways very early on. And what we can do is we can sort out potential liabilities, that's the key phrase, or potential hazards, very, very early on before you invest millions of dollars into it. We can advise the drug development teams on what these potential liabilities are, and they can decide at that point whether or not to pursue that molecule or look for another one. What is the target? I can tell you from personal experience, advising on some of those teams, we have saved, or if they had listened to me, they would have saved about $16 million. And that was just for one compound that they decided to go forward with, even after my advice. So, by the way, they got in trouble for that decision also. They were held accountable. So once you get into a little bit more towards the end of that discovery phase, when you're getting really down from like the millions of compounds down to the thousands and hundreds, that's when you're really getting into that lead discovery time point. You start getting more and more into the in vitro assays. Screening, trying to look at structure, activity, relationships, particularly in relation to known liabilities for genetic toxicology or interactions with endocrine receptors, things along that nature. And all of that can lead you to whether or not this compound has a potential liability again. It's not definitive proof. These are not pieces of data that you are going to stake your entire, submission package to the FDA on. But what it will do is it will supply your drug development team with information on directionality to go within your compound series. A little bit later, you get into what is, again, most people refer to as the preclinical evaluation. Other people refer to it as your IND enabling package. And essentially, this is where most people think toxicology lives and dies. Not true, but this is what most people will think of it as. And those are your GLP regulatory studies, which support your first time in human clinical trials. And an acronym that you'll hear a lot is FTIH. It's your first time in human. It's your phase 1 clinical trial. All the same thing. So in general, you have your single dose, your short-term repeat dose up to, depending upon the nature of the disease you're looking at, up to usually about a 28-day tox study. Your safety pharmacology and genetic toxicology will go into that, as well as a whole host of other compound specifications and efficacy studies leading up into it. Going further into, you're now in that clinical space. Toxicology is still going on. In fact, it is stronger at this point with a lot more activity going on than in preclinical. Now you're getting into your subchronic and your chronic toxicology studies. You're getting into the reproductive toxicology. So subchronic is pretty much anything less than about 6 months. Your chronic is anything a little bit longer than 6 months. And within that range is you then start getting into your carcinogenicity studies, which are for 2 years. So very long-term studies. And that continues on until you're out to the market. I'm going to actually show you what a typical drug development plan will look like. When I show it to you, understand this is a quote-unquote standard. I have never run into a standard drug delivery package yet. But you have to start somewhere. So even after it goes out onto the market, though, toxicology still goes on. People are being given this drug. Question: how many animals do you think in all of the toxicology studies combined, how many animals receive that compound? Okay, you spoke up. I got to hear it more now.
Too many.
Too many animals. The answer to that is yes and no. 1,000. It's probably closer to about 1,500 to 2,000, depending upon the breadth of the studies you have to get to.
And it's just a GLP one.
Those are the GLP ones. Those are not efficacy studies, any of the pre-work that's going on. How many people are going to see a compound?
Phase 1 or
Post-market.
post-market? Oh.
It depends. So you have anywhere from your orphan indications, about a few hundred, up to millions of people. Genetic diversity amongst people, are we inbred or outbred? I'm not talking about West Virginia. Sorry to anybody who's from West Virginia.
We are in Alabama.
I wasn't going to say it.
You're smart.
So we're very outbred. We're very genetically diverse. Toxicology species, genetically diverse or not?
No.
Not. What are the chances then of you seeing an adverse event or a toxic signal once you're out into the human population that you didn't see in your toxicology studies? Big chance, little chance?
Yeah.
Hopefully it's a small chance, but it does happen. So that's why you will hear about all of these compound registries. And that's so that when the product's out on the market for the first few years, if you have something happen, your doctor needs to know about it, your doctor reports it to the FDA goes back to the company, and then we have to do additional testing to try and discover why didn't we see that, why didn't we know about it, and is this idiosyncratic, meaning it's one-off related to an individual and not to the compound drug, or is this a real finding that really could impact the population at large?
So how do you test combination of drugs?
I'm sorry.
How do you test combination? Let's say you have your compound and somebody is taking, I don't know, high blood pressure, medicine, and the two interact with the toxicity.
So there, from a toxicology standpoint, we don't test that directly, but you do have in the pharmacology world, particularly the ADDA, they do a lot of drug-drug interaction studies. And if you're seeing an individual that actually has what we hope is an idiosyncratic effect, we get their history from that, we find out what drugs they've been taking, and if it hasn't been on the list that we've specifically tested, we can go back and test that.
But in fact, you have to wait till the case happens.
Yes, we do. Yep. There is no way possible for us to test every single combination that's out there. They do a pretty good job at trying to look at metabolic pathways and trying to sort out what types of compounds go down a particular metabolic pathway. And with that understanding, do we believe that it will interfere or not? And then use exemplars of those to test against. But it's not perfect because you can't test everything.
Any idea of the percent post-approval marketed drugs that get formed because of toxin use?
Percent, I don't have a percent. I know of incidences that it has occurred. There are more instances where it's happening within the past 10 years than I saw prior to that. Part of that is an increased vigilance and sensitivity to those effects. If you go back and look at really prior to, let's say, 20 years ago, toxicity was not seen as that detrimental with a lot of drugs.
It's cost of doing business.
Cost of doing business. Aspirin, safe, not safe. Depends on the dose. It is also a teratogen.
Oh.
It is, quite frankly, the only reason why it's on the market now is because it was approved decades ago. If it were to go through drug development in the FDA right now, it would probably fail from a safety standpoint.
At one point, you like to measure the benefit versus
Yes.
the toxicity. I mean, are you going to, are the fields going to trash many good drugs just because the tox profile is bad, or are you going to look at something more in the middle
Do
ground?
you allow one person to die so that 1 million people can not suffer a headache? I mean, those are the decisions that have to be made.
The problem is there is not going to be any drug.
results.
Exactly. It's a very fine line, and it's a balance that people, you have to walk it every single day.
Yeah.
Risk-benefit ratio is the question when it comes to drug development.
Mhm.
The nature of the liability is a factor as well. Is it something that is treatable? Can we take them off the drug? Will they recover from it? Is a permanent lesion that will occur? How debilitating is that toxicity? All of that goes into it. Nothing here is really black and white. That's why it's really difficult to manage. It's also why you see compounds that actually get out to the market, because we believe that anything that we've seen is something that could be managed. What can't be managed, with one in particular, are the cardiomyocyte necrosis which occurs. Not seen in the actual tox studies. It was starting to happen in people, not manageable. It was yanked. Okay. Going on. Discovery phase. This is where you do a lot of the in vitro work. I'm deliberately putting a lot of things on here because as a toxicologist, I'm very tox-centric, but we don't work in a vacuum. We have to work as a team with all of the chemists and all of the biologists. Quite frankly, we can't do our job if the chemist actually doesn't manufacture the material for us to test. We can't actually do any of our studies or analyze them if the pharmacy and the bioanalysis team actually doesn't have a method in place to detect it. Okay? So I'm not going to go over everything in these slides. These slides will be available if anybody wants them afterwards. But in general, you can see candidate, oh, by the way, candidate selection is just it's slang in the industry for, that's when you get to your lead compound that you really have confidence in and you really want to start progressing it into true development phase. Okay? So going from your lead to you have confidence in one of them. So a lead, you'll probably have anywhere from 5 to 10 compounds that you're taking into this phase. And then candidate selection, you're down to one that you have faith in and you're going to progress it further in drug development. So you do a lot of your genetic tox screenings, non-GLP at this point, a lot of the investigative work. Frequently called in vitro toxicology. However, I also run across a lot of people who think that in vitro toxicology is the same thing as cytotoxicity. That is a component of it, but in vitro toxicology, again, covers a much broader area. The whole point of it is to provide information on the mechanism of the drug. It's an indication of events. Human body, how many different cell types are in it? I don't know.
10,000.
But do you think that HeLa cell in vitro is going to predict everything that's going on in your body? No. What it will do, though, is it can find a bad actor in a compound. So if something is very cytotoxic, you'll be able to pick that up, basically take that compound off the shelf, put it in the garbage can, not go back to it. Okay? That actually helps the chemist, again, determine which portion of the chemical series they want to go after. It helps refine your chemical library into those that are potential drugs and those that are just, toss them away, never look back. Says here, in vitro methods widely used for screening and ranking chemicals, studying cell tissue or target-specific effects, improve subsequent study design. Absolutely true. Less expensive than in vivo studies. True or false?
Yeah.
True.
Well, depending on the shape.
It depends.
Yeah.
I do some in vivo studies that are actually cheaper than some of the in vitro
Yeah,
studies.
that's true.
Faster than in vivo studies. True or false? Depends.
Yeah. Usually.
Same thing. Well, it depends on that.
Yeah.
If you're looking at some of the short-term tox studies,
Okay.
you're in and out within a week. Some of the in vitro studies, you have to get the cells up. They're going to grow for 3 weeks before you can use them. Then they're going to be treated for a while.
Yeah.
So depends on the studies. So a lot of these sort of like rule of thumb reasons for why in vitro is, so fantastic, we have to stop doing animal work to go over to it. It's a yes and no. There's a balance here as well. In vitro is compliant with the 3 R's. Replacement, reduction, refinement. Okay? One of the things that I am a huge proponent of, my life, my career has been in vivo toxicology. Only do it when you need to. Use the appropriate model. If you don't need to go into an animal in order to get your answer, don't go into the animal. Do your science ethically. Okay? In vitro is somewhat less predictive than your animal models. It's true. Does that mean animal models are greatly predictive?
So how are you addressing this? Because there's a big difference between a lot of the model organisms and humans in pharmacology, metabolism, the reaction, so toxicology. How do you take that into account?
Okay, so you're stealing my thunder later. Yes. That right there is the reason why you do things in multiple species. That is also the reason why you have computational toxicologists and why we're looking at things more on a molecular level and a pathway level in trying to determine what is the similarity and difference between the animal model and the human. So all of that is now being taken into account. And all of that is also the reason why drug development costs so much. Anybody know the current amount on getting a drug from conception to market?
1.8 billion.
Very close. Current estimates are 2 to 6 billion.
It's time that they show some money to me.
You don't get a commission off of it, unfortunately.
No, for my research.
Okay.
You think it justified or is overdone?
I'm sorry?
You think they justify this amount of money or they're overdoing it?
That's a good question. I think that they are overdoing it in many cases.
Yeah, it's what I did. I don't have the,
Yeah.
I don't have justification, but I believe they're overdoing it.
A lot of the regulations are there very specifically because of bad actors, whether that is a bad actor is a chemical or whether the bad actor is a scientist.
Yeah.
So a lot of the regulations are in place to make certain these things don't happen again. But what it means, though, is you have layers and layers and layers of work that have to go into it, layers and layers of regulation and documentation in order to make certain that you as a scientist are not fudging anything. And all of that costs money. Just taking the same study from a non-GLP standpoint to a GLP, where the only difference is additional layers of documentation, science is the same, study design is the same, results are the same, just different layers of documentation. For a $100,000 study, you make it GLP, it's now $150,000. It costs. All toxicology studies that go towards an IND submission, investigative new drug, is required to be GLP.
Yeah.
So every single one of the studies, you're now adding like 50% on top of.
Yeah.
It adds up. In vitro toxicology. Most people think of it in that little orange area on the scheme of things. But again, it is actually more expansive than that. In vitro toxicology, in vitro models are important very early on at target validation stage. And you can get potential efficacy or proof of concept at that point on basically even just the target that you're going for, and some preliminary toxicology data. When you're out in your actual clinical trials and something happens to an individual and you're saying whether or not this is human related, did we see it before, why didn't we see it in people, there are models out there now where you can actually take human explants and you test in humans. At that point, tissue culture, but still in humans, to try and sort out mechanism of action. Is this real? Is it idiosyncratic? So again, all across the gamut. So that's where most people think it resides. There's a lot more out there. In drug development, this is what most big pharma will do A lot of small biotechs, they don't. The reason for it is a biotech company is trying to get the minimal amount of information to then divest and have somebody else buy it, who will then go and do a lot of this. Okay? And this is along the lines of predictive toxicology. It's before you actually get into your IND submission. They're generally done at the same time as candidate selection, and sometimes right alongside of your GLP tox studies. Is it necessary according to the regulatory bodies? No. Do we do it because in the end it saves us money? Yes. Again, what you're doing is we call it early attrition. When is it better to fail a compound in drug development? Early or late? Early. Clinical studies cost a lot of money. So if you're going to find something, find it early on. So pharma will invest early on to do these non-essential tests to determine which compounds really have a strong likelihood of becoming a drug in the end. And they start looking at secondary pharmacology. What is secondary pharmacology?
The activity of predictables?
All the way in the back, by the door. Nope. Back.
Activity of imitables?
Possibly. Any other guess?
I'm an effect in a different target that the drug's supposed to hit. I don't know.
Absolutely correct.
And Claudia, we're going to
Yes.
make you hire myself.
Okay.
Yes, absolutely correct. Your drug development team is going to come up with a chemical or biologic, and they're going to come to you and say, this is specific, it only hits S1P1, nothing else. They're lying. Well, they may believe that, but it's probably not going to be true. Just like with toxicology, it's all in the dose. With your interactions with other targets, it's all about, again, the dose. I will put money on you bring me any compound, it's going to interact with more than one other molecule. Okay? That can have side effects. All the computational toxicology in the world, based off of what your intended target is, it's not really going to be that critical when you get into an animal where it actually hits 20 other things. Tyrosine kinase inhibitors are notorious. We call them promiscuous. One of them I was working with actually hit 20 other targets. The team told me it was specific. When you started to, diving into it's actually what it binds to otherwise, it really hit. It was at least 20 other kinase receptors. And you had all of these side effects determining, okay, well, the team kept asking me, well, is it target related or not? Which target? In the end, we had to scrap it. And instead, we went forward with a compound that actually only interacted with 10 others. It was better.
So I have a question. So
Yes.
do we think that some targets maybe will not be too much efficient because of the secondary? What I mean is that is it the target, the best drug will be a drug that really goes into a single target with not other?
Not necessarily, no. We have found, quite by serendipity, that a couple of compounds that were going after one, again, this was a junk kinase. When we hit only one of them, and by hitting only one, it was because we reduced the level to where the binding interaction was almost sufficient for, based on the affinity with one of them, so it didn't hit anywhere near the IC50 score or lower. It really wasn't effective. It was only after we hit 3 of the junk kinases that it truly became effective.
But you keep this drug or you?
We kept that one.
Yeah. Okay. So basically the assumption was only one hit, but in fact it worked really because you did, for
Yes.
classes. So
The initial assumption was good in theory, didn't work in reality. So we actually needed to branch out and hit multiple targets for it to function. from a disease amelioration standpoint.
So when you say predictive toxicology, so you are running a computational simulation?
Part of the time. So some of this is done in silico. The early safety predictions are done completely based off of information that's already out there in the literature. The in silico is a computational model.
But when you say
A lot of the safety pharmacology, genetic toxicology, those are in vitro assays, which are done sometimes. Some of them are, you're looking at the structure and you're looking at large databases and trying to find similarities in structure with known liabilities.
when you say a kinase is, I mean, the developer tell you, I mean, it's specific, so they must have run a profiling experiment telling you, you know, it's only targeting particular kinase, and then
Yes.
you found out, I mean, there's 20 extra, I mean, this is based on simulation by docking or something?
No, that actually wasn't simulation. Those were real binding assays which were done. Again, this is a difference in perspective between a biologist and a toxicologist. A biologist is looking at this and saying, I have a 10-fold increase in selectivity with my target compared to everything else. Therefore, it is selective. And they don't necessarily tell you about everything else it binds to. So as a toxicologist, we know that those other things it binds to can be critical.
Yeah.
So we have to go and investigate. And we have to look at all of the binding data and see if there's an issue or not. And quite frequently, in my experience, a lot of the toxicities we see are associated with the secondary pharmacology of the compound, which was not necessarily advanced or highlighted sufficiently early on. Secondary pharmacology targets. The industry has actually come together pretty good, and there's no way in the world we can test every single molecule that's out there, so they pick sort of key players in different arenas, whether it is related to cardiovascular function, CNS function, renal function, and they've identified this set as fairly representative. If you start really affecting any of these, you're going to have a liability, probably. I say probably because sometimes, really in particular to your ion channels, depending upon how you hit them, they can compensate for one another. So that's also one reason why your in vitro toxicology is not necessarily what you want to bet all of your money on, because you can actually have a sodium channel blocker that actually affects your calcium channel, and in vivo they will compensate for one another and there is no physiologic response to it. So it's not an issue in a person, but in that individual cell, when you're doing the patch kind of assay, it's a huge effect. So generally what you will do is you will look at, I'm just using ion channels as an example, you'll look at the ion channel data from the in vitro assays, and then you will find your bad actors, and then you'll do a quick in vivo study to confirm whether or not there's any in vivo relevance to it.
So let's say that you are not a big pharma like stuff, you know,
Yes.
and you want to do those kind of assays. So can you gains companies that are doing that? I mean,
Yes.
this is a huge work. I mean, I'm thinking
There are actually a large number of contract research organizations out there that will do all of this work for you. There are companies now who are doing drug development that are basically virtual companies. You have two scientists that talk over the phone, one in California, one in New York, and they will come up with a chemical. They will farm out having the chemical produced. They will contact CROs and have them do all of these studies for them. And this actually does occur in real life. So you actually don't have to have the facilities yourself to do all of it.
But I believe this kind of assays are very expensive.
Yes, they are. Most small biotech will try to divest it before doing this, and they will really rely on the pharma who buys it to do it. Small tech versus big pharma. Small tech has a higher risk threshold, and they really want to simply get their product, this is a gross generalization, they want to get their product to a sufficient advanced stage where they can sell it. And they're going to sell it to somebody else who can do further development and get it to the market. They don't do as much risk analysis as I would personally like. They don't do as much efficacy testing as I would like. They will do a quick proof of concept, and they do what I, in my jaundiced view, think of as very bad science when it comes to the toxicology. They will go in at an obviously low dose to say it's safe, and they will try and sell it. One of my jobs at GSK was to actually review potential in-license opportunities. And looking at their tox packages, they were basically worthless as studies for us to determine anything off of. We would have to start back at square one. But they were declaring to their investors that it was safe.
Raising money.
They were raising money. So, I mean, there are reasons for it. When it comes to the differences between them, these small biotech companies do a fantastic job at discovery. It's the development that they don't seem to do that well. The goal of all of that work, though, is to get it to a point where you actually have better compound selection before you ever get into the animals, before you ever really start spending the big bucks. Just so you know, again, general prices, your early, like, let's say rat dose range study is going to cost about $40,000 to $60,000. A monkey will be $100,000 or more. Your 1 month tox study in a mouse will be anywhere from $300,000 to $500,000. In monkey, you're talking about close to $1 million for that study.
Yeah.
They're not cheap. You want to cut your losses before you get to that point. Okay? So a clinical trial, a very small clinical trial, your first time in humans, typical cost is going to be $16 million. Again, cut your losses before you get to this point. So in vivo, you've done as much pre-work as you can. You've done as much outside of the animals as you can. You now have to get into the animals to get real tox data out of it. You want to establish a safe starting dose in the clinic. There are metrics for translating with adding precautionary bumpers in there from your animal data to the humans. Again, this is another reason for going into multiple species. Believe it or not, I'm not a rat. Or I'm also not a dog, or a rabbit, or a ferret, or a guinea pig. Those are what we do the testing in. But you have something that's called species concordance. If you're testing something in multiple species and you're seeing the same signal, chances are you're going to see it in humans. If you see a liability crop up in rat, but you don't see it in dog, it's still a potential, but it's less of a red flag. You can still proceed, you just do it cautiously at that point. Okay? So you also want to try and define what potential target organs are. Again, you're going to go into the clinic. You want to make certain that in the clinic you have biomarkers in place for it. You want to, is this trackable? Is it reversible? That's all of this work that you do before getting into the clinic. So here we go. This is your GLP IND enabling package, standard. You have your GLP genetic tox work, your AIMS, your mouse lymphoma, your micronucleus. There are a lot more assays that can be done. There are a lot more that are done. These are the basic ones that need to be done. Okay. Safety pharmacology, your rat Irwin. Does anybody know what an Irwin study is? It's a very specific study type looking at potential CNS effects on behavior. Rat respiratory study, your cardiovascular study, which is usually done in a non-rodent, but can be done in a rodent, can be done in a non-human primate. We try to go into dog. Dog, believe it or not, seems to be the most translatable for CNS effects from the animal to the human. Not for tyrosine kinase receptors. They're very, very exquisitely sensitive to those. So if you're done with those, probably dog isn't going to be the best bottle for you. Then you also have your repeat dose tox studies. Okay. That goes on. You're getting into, you're done with your first time in human. You're moving from your phase 1 clinical trials into what we consider more proof of concept, your phase 2s, your 2A, 2B. That's when you start doing your reproductive toxicology work. What sort of clinical trial do you do where you actually go in and test for teratogenicity?
It involves females?
You don't. You never go into human trials in a pregnant woman.
Oh, yeah.
You just don't do it unless the indication is trying to, for preeclampsia. But that is a massive exception, and there have to be so many regulatory guidelines and safety measurements put in place. It's an extremely difficult, extremely expensive study to run because of that. That's why these are highlighted in yellow right here. These are critically important to drug development. If something is labeled as a teratogen, chances are automatically 50% of your population cannot see this compound. Potentially more. Is the drug passed in semen? That's going to have to be determined. You are not deliberately going to go and test and see if I can cause a malformation in your baby. It's just not going to happen. This has to be done very, very well. The data you have to rely on. Most of the other tox work, yes, you do test it in vitro, you test it in animals, but you're also going to be testing it in your clinical trials. You're not this. The only way you're going to get human data out of this, 99.999% of the time, is after it's been on the market for years and your compound registry starts seeing a signal that comes up. That's not something you want to see. Sure, everybody's heard of thalidomide. It passed through all of the non-clinical toxicology. It was only after it went out into the human population they found that there was a real problem. So also during this phase, you're getting into your subchronic, your 3 and 6 months, and you're starting the early pre-work for your carcinogenicity studies. Okay? So your carcinogenicity study itself is 2 years, but you have to do a dose range find and stick before then. You've got to do a subchronic study before then. And all of it has to be backfilled. So you're actually looking at 3 to 4 years before you need that data for you to start on all of this. From your proof of concept to market, this is around your phase 3. Okay? You're now doing your more chronic tox studies. This is where you're doing that carcinogenicity study, not preparing for it. This is where you're also doing your 9 to 12 month tox studies. Why do we do these for so long? And do you have to do that on every single or for every single drug?
I would say no, because these drugs that we need 2 or 3 short term, and some that, for instance, we are taking an aspirin, we can have aspirin a day.
All of this, and the reason why at the very beginning I used the air quotes, which I think is so pretentious, about this is a standard design, is because all of this needs to be matched to what your clinical, what the clinical trial is going to look like, and how it's actually going to be used once it goes onto the market. For Benadryl, I use it every single day. They have to go out and they have to do multi-year studies to make certain that with continued administration, you don't see effects that crop up later on. For an antibacterial that you are going to use in the hospital, so you're going to get it for 2 weeks, and then you stop and you go away. What do you need to do in that? Do you need these 9-month and 6-month studies? Actually, that was a trick question. That was a trick question. The answer is yes, you actually still do. But not to get into clinical trials. But when you start getting closer to market, you are going to have to do those. So So with the clinical trials, what you have to do is you have to match almost a day-for-a-day basis on how long you're going to be in people with clinical trials. However, particularly with antibacterials, there's potential that you're going to have it or use this repeatedly throughout your life.
Yeah.
There's potential that you're going to get infected again and again and again, hopefully not, but potential. So they have to look at what extended durations will. So you would still have to go there. Probably not the 12 months, but 6 months I would put money on you have to do. Even if in reality you're going to see it for 2 weeks. Post-marketing. I'm telling you, the talks never ends. The chemistry never ends either. There are line extensions. There are changes in formulation. Oh, by the way, your compound is not the only thing that we're testing. It's actually how it's formulated. So if you, as a drug developer, say, okay, I just want to change the formulation, we're just going to go for it anyway, it doesn't work, you've got to go back. You have to do the toxicology in that formulation.
So you have to do all again?
We do piecemeal work, we call them bridging studies. So you can go in, you can get a bit of information about the toxicokinetics. Does it change the actual exposure profile? Does it change the metabolism? And then you will do a few select tox studies to determine whether or not anything's changed from the tox profile. You don't have to do the complete package, but you do have to go back and do it.
A question. So they say that you would like to do an IP. So all the animals that are being the toxicology.
And so if you're very soft-spoken, then that's very
Yeah,
I can't hear.
so let's say that you said that your new drug is the IV.
Yes.
All the toxins animals actually given IV.
Does it have to be given?
Or can it be IP or is that the easier way?
The FDA very much prefers that it be given in the same route that you're going to give it in humans. There are some exceptions to that. One of the exceptions are the teratogenicity studies, and that's because what you're really looking at there is the effect on the developing fetus. So whatever the dose route is, it's the exposure to the fetus, so you can give it pretty much any way you want. If you have difficulty in formulating, that would be acceptable for your animal model, but is in humans, you can still go forward. But there is a larger burden on you to actually define any potential differences that may come up from that. So there's going to be additional work that you have to do on top of it. It's not a no-go, but the hurdle is higher and more difficult. Okay. Other things. See, I told you, I had it in red here. Why the two species? It's still my letter. Drugs for life-threatening illnesses require fewer studies to reach the clinic. Why is that? I'm a cancer survivor. Quite frankly, it was my life on the line. I didn't care if I lost my hair at that point. I care now. But at the time, I didn't care. A lot of the toxicities. I had second-degree burns on my body from all the radiation. I didn't care. It was saving my life. Life-threatening diseases have a much lower threshold when it comes to that. And it is because what is the risk versus the benefit? The benefit is you're not dead. You're going to accept a lot of potential liabilities on top of that. Now, years later, people come back and they're upset because, real example, saving a lot of men's lives, but it made them sterile. And the fact is, you're still alive, but you can't have children. They were not necessarily informed when they were given the drug that that was a possibility. So from a liability standpoint, the company was liable because they were not properly informed. However, did they take this off the market? No. They put in the consent forms now that this is a very real possibility. People make that choice going into it. A lot of times, and what their company is actually offering is that they will bank sperm for them for free as part of that. Other species, the number of toxic studies, it does differ depending on it. We've already gone over that. Key assumptions, other organisms can serve as an accurate and predictive model for toxicity in humans. The reality is the animal models are predictive depending on a lot of factors. They're not humans, okay? So the general percentage is the best of the animal models is about 80% predictive. That means 20% of the time they're wrong. Again, that's why you do multiple species. You're hoping with the different species that you're catching different aspects of it, so then your overall prediction or predictivity increases to, let's say, 90, 95%. The in vitro assays, a really good, strong in vitro assay is about 70% predictive. Understanding the strengths and weaknesses of any particular model, absolutely. If you have a tyrosine kinase receptor inhibitor, probably not a good idea to go into dogs. Okay? Caveats. Drugs showing safety and efficacy in preclinical animal models may show very different pharmacological properties when administered to humans. Yeah, they're not human. Okay. Development of proper preclinical models could efficiently predict drug behavior in humans is essential prior testing in a human subject. Is it essential? It's a good idea. It's a really good idea. I have known very successful compounds that didn't do any of it. They went straight into the, believe it or not, they went straight into humans. This is about 100 years ago. But it worked. They didn't do animal models. Has anybody heard about the Poison Club? Okay. The very first, in the United States, the very first actual tox program, they did testing in humans. It's called the Poison Club. They got a lot of volunteers. There were a lot of manufacturing and environmental materials that the government was curious about. You know, is it safe? Is it not safe? So they hired a bunch of volunteers to come in and eat these compounds and to see over time whether they're going to sick or not. It's called the Poison Club. True story. The FDA and other regulatory agencies are more and more requiring sponsors to provide data to support selection of the specific species. Absolutely true, especially in relation to the use of non-human primates. Rule of thumb, rodent, yes. If you're going into non-rodents, there's a tiered approach where you have to address each one of the tiers before you go on to the next. Going into dog, that is the default. If the dog is not an appropriate model, you have to explain why. Then you can go into mini pig. If the mini pig is not an appropriate model, you have to explain why. Then you can go into non-human primate. And you have to explain why that is suitable. The future of tox. I have like 2 minutes left. Tox is expensive. Drug development is really expensive. The chance of success is minimal. This is big money business. The reason why your drugs cost so much is because of the price of developing it. And essentially you have to patent it. So you have to, in the patenting the compound, so you get a 20-year patent life, about 10 years before it ever gets to the market. You have to patent it to protect it from competition. So you only get, if you're lucky, 10 years of revenue, uncompeted revenue off of that. And that's after spending billions and millions of dollars in trying to develop the 10 compounds which didn't work to get the one that does. They're really looking at toxicity profiling and trying to figure out where you can do better attrition to cut compounds early instead of late. The main reason for failure in the industry is toxicity. You've got clinical safety, efficacy, and then you have toxicology. If you combine toxicology with the clinical safety, safety is the biggest reason for failure on compounds, followed by efficacy. Looking at one particular company, at their work, clinical safety, clinical efficacy, again, pretty much reflected what's going on with the industry at large. If you're looking at what are potential target organs that you really need to pay attention to, here is a sort of a histogram related to failure of particular compounds based off of toxicities with particular organ system. Cardiovascular being the biggest one. Why? You can't really rescue somebody if they're having cardiac necrosis. It's not reversible. Liver damage, depending upon the nature of it, it's largely reversible, but not always, and can be life-threatening. Central nervous system effects, things like that. Those are critical ones. Then you get down to like the eye, the lung.
Death is one side.
Death is one of them, yes. That one's not recoverable, usually.
Mostly death.
Mostly dead. So a large part of this is the lack of translatability, that 70 to 80, if you're lucky, 90% predictivity from the animal models into human. That's the real root cause of it. So the future of toxicology is actually going in a different direction, and that is to put in what are considered complex in vitro models into that overall drug delivery scheme. So not your high throughput screens. These are going to be, at best, medium throughput, more than likely low throughput. They're complex, multicellular in vitro models that the plan is to use human tissues from. The real future goal is to be able to have banks of different geographic areas, different genetic backgrounds of stem cells from people, and use inducible pluripotent stem cell technology to create the different models that you want. So then you can say, if we're going to go over and into sub-Saharan Africa, let's actually test with the general population that's over there. What's their genetic background? If we're going into India, there's a different genetic background that's over there. And you're going to test to get a better understanding of the potential liabilities that are in the specific geographic area. That's in the future. We're not there yet. These models are still in development. There are huge initiatives going on right now. Some of them are funded by the NIH and NCATS. Forget what that actually stands for.
National Center for Advancement of Translational Science.
Yes.
They funded CCTS, which paid for your lunch today.
Very cool.
Thank you very much. Again, if you haven't signed in yet, on your way out, please sign in. Any questions? I think we had good interaction, Drew, and we opened the chat bubble just in case.
I heard some things along the way.
And we just kind of like, yeah, there's no chats. Okay. All right. Well, thank you everybody for coming. Next week, we're going to talk about clinical trials. And hope to see many of you then.
Yes.
Thank you.